Hello, everyone. We welcome you to our journal club today, where we have Dr. Jazmin Calyeca, who is a research scientist at Nationwide Children's Hospital in the Department of ENT Surgery and Center for Regenerative Medicine. She presents her research titled Orchestrated Response from Heterogenous Fibroblast Subsets Contributes to Repair from Surgery-Induced Stress After Airway Reconstruction. She is joined by Dr. Debra Boyer, who is the Chief Medical Education Officer and Designated Institutional Official at Nationwide Children's Hospital, where she also serves clinically as a pediatric pulmonologist and physician on the lung transplant team. The moderators today include Dr. Elaine Dye, Director of the Sarver Heart Center Data Science Unit and Translational Scientist, as well as myself, Tu Tran, neonatologist and Translational Scientist at UT Health in Houston. We invite everyone to enter their questions in the chat, where we will monitor or wait until the end of the speaker's presentation to unmute their microphones and join in for a robust, engaging discussion. Go ahead and take it away, Dr. Calyeca.
Thank you so much. Good afternoon, everybody, and thank you for the kind introduction and also for the opportunity to present here today. Before I begin, I would like to acknowledge all my team members and especially my mentor, Dr. Tendi Chan, for all the support during the development of this project. Here is just a brief outline of today's presentation. I will start with a very short introduction, and then I will walk you through our approach, the main findings, and finally, we will discuss some of the clinical implications of this work. I will begin with the basics for everyone. The trachea is also commonly known as windpipe, and the main or primary function of this structure is to allow passage of air to and from the lungs. Medical conditions that can affect this function or are obstructing the airway can result in breathing difficulties, and some of them require surgical intervention. Airway reconstruction is a surgical procedure used to restore a damaged or narrow airway and can be performed after congenital abnormalities, injuries from trauma, or also to remove scar tissue. It is also an essential step developed during lung transplant, and as any other surgical procedure, it carries several risks, mainly at the anastomotic site. However, for the purpose of this presentation, I will focus only on stenosis and fibrosis. Effective healing requires the proper response from the airway structure. The inner lining of the airway is formed by a pseudo-stratified epithelium that has several types of epithelial cells, the submucosa area that can have blood vessels and nerves, and below the cartilage. In normal conditions, these structures are well organized. After airway reconstruction, we are expecting remodeling in the submucosa area as part of the proper wound healing. However, if this process is deregulated and there is an accumulation of excessive collagen, mainly can result in pathological scarring and also narrowing of the airway that then result in obstruction. Our group and others have tried to understand what are the conditions that allow proper wound healing versus the one that result in anastomotic complications. We propose that airway reconstruction triggers a surgery-induced stress response, and that the response is deregulating the cellular communication in the airway tissue, and if uncontrolled, can promote the anastomotic complications, mainly stenosis and fibrosis. To prove this, we create a preclinical model by transplanting a tracheal graft into an identical genetic host. And we use that as a surrogate for airway reconstruction. And we evaluate how the surgery-induced stress affect airway homeostasis over time. We did that at 2 selected time points, at day 14 and day 28, that we call early repair and late repair. And we evaluate both clinical and molecular aspects. After airway reconstruction, we saw that the submucosa area was mainly an acellular matrix with a few blood vessels and cells. However, after airway reconstruction, we saw that the submucosa area had active remodeling and presence of infiltrated cells that result in the thickening of the submucosa. After seeing that different cells were infiltrated into the submucosa in response to surgery-induced stress, we then ask us which cell types are infiltrating in that area and what are the cellular processes that they are activating. To answer this question, we use single-cell RNA-seq. That is technology that allows to evaluate the gene expression at cellular level. In brief, what we did was collecting airway tissue, and then we generate a single cell suspension, so we were able to see each of the cell types of the airway. We then barcode each of these cells, so we were able to see the gene expression, and finally use deep computational analysis. With this computational analysis, we can represent each of the cells as a dot, and cells that have similar transcriptomic signature will cluster together, suggesting that they are part of the same cell type or identities. Using this technology, we were able to identify main airway cell types, including the different parts of the epithelium, like club cells, basal cells, and also fibroblasts, endothelial cells, and chondrocytes. One of the things that was interesting for us is that we saw that in response to surgery-induced stress, fibroblasts were the most abundant cells, and they were drastically increasing. We then use immunofluorescence to validate the findings from the single cell. And we use one of the specific markers for fibroblasts, that is pimenting. We basically saw that in normal conditions, there were a few cells in the submucosa. However, our single cell suggested we have infiltration of fibroblasts in the submucosa in response to surgery. And that levels and numbers of fibroblasts were correlated positively with the collagen content found in the same area. After identifying that fibroblasts are mainly infiltrating into the submucosa, we then ask us if these fibroblasts were a single population or they were formed by different subtypes. So we came back to our single cell RNA-seq data, and we use again computational analysis to try to answer this question. Using specific markers, we identify 5 clusters forming the fibroblasts, and we name them accordingly with their function and the gene expression as CTHRC fibroblasts, adventitial fibroblasts, perichondrial fibroblasts, immune recruiting fibroblasts, and finally, airway fibroblasts. We saw that in homeostasis, adventitial, airway, and perichondrial fibroblasts were the most abundant cells, and CTHRC and immune recruiting were almost absent. However, after airway reconstruction, we saw that the number of these cells changed, and the most abundant cells were CTHRC fibroblasts and immune recruiting, although the perichondrial fibroblasts remained there. We could see here how surgery-induced stress was shifting the proportion of fibroblasts. After identifying this new subpopulation of fibroblasts in the airway, we then asked us what was the function. Fibroblasts are known as collagen producers, but we wanted to see if that was the only function that they have or they have more specific functions in the airway. So we began studying the perichondrial fibroblasts. We came back again to our single cell RNA-seq data, and perform another type of computational analysis called pseudo-time. This analysis allowed you to identify the cell fate of the cells. And we saw that during normal condition, perichondrial fibroblasts have a balanced response and have the same commitment to either self-renew or differentiate into chondrocytes. However, after airway reconstruction, the perichondrial fibroblasts are only committed to differentiate into chondrocytes. So that was suggesting that the perichondrial fibroblasts have the ability to differentiate into another cell type. We then moved to histology, trying to see if what we identify in the transcriptomic analysis was true. And what we saw is that during normal conditions, we saw the appearance of some structure that have this fibrocartilage shape. And after airway reconstruction, the size and abundance of this structure was increasing. However, it was still not confirming that those cells were indeed fibroblasts and chondrocytes, or fibroblasts differentiating into chondrocytes. So then we use immunofluorescence to validate that. We perform that technique using both fibroblast and chondrocyte markers. For the fibroblast markers, we use collagen 1, and for the chondrocytes, collagen 2 and SOX9. And we saw that after airway reconstruction, the structure that we saw in histology were indeed cells that were expressing both markers, confirming the capacity of the perichondrial fibroblasts to differentiate into chondrocytes. We then move with the next cell type that we identify, the immune recruiting fibroblast. This population, we name it like that because one of the markers was different cyclins that participate in recruiting immune cells. And we saw that the number of this population was exponentially increasing after airway reconstruction, starting from 1% to 13 and 47% after airway reconstruction. Trying to understand the function of these cells, we did another computational analysis, and we took basically all the genes that were deregulated and asked where were the function of those cells, of these genes, sorry. And what we identify is that most of these genes were participating in regulating the immune response, as we expected. But important for us was that we saw that they were particularly attracting neutrophils. So we then used immunofluorescence and stain for neutrophil markers, and we confirmed that after airway reconstruction, neutrophils were accumulating in the submucosa area, in some way supporting that the immune recruiting fibroblasts were actively attracting neutrophils. We finally moved to another of the identified cell types after airway reconstruction, the CTHRC fibroblast. And the CTHRC fibroblast was the most abundant cell increasing in numbers in response to surgery-induced stress. However, the presence of these cells was transient. It was higher at day 14, but then we saw that at day 28 was slightly decreasing. So we then used immunofluorescence, trying to see if the same was happening in vitro, if we were able to corroborate this transcriptomic data. We use again a specific marker for fibroblast, like mimentin and CTHRC. And we saw, confirming our transcriptomic data, that activated fibroblasts that express CTHRC were accumulating in the submucosa area in response to the surgery. But at day 28, they were slightly decreasing in numbers. So then we explored more about the function of those cells, and we saw that among all the other fibroblast subpopulation, the CTHRC fibroblasts were, the CTHRC1 fibroblasts were the ones that expressed more collagen genes, suggesting that their function was focused on remodeling, specifically on producing collagen. Together, these findings reveal a coordinated but imbalanced fibroblast response. We saw that perichondrial fibroblasts can contribute to cartilage changes. Immune recruiting fibroblasts drive neutrophil infiltration, and CTHRC1 fibroblasts can promote collagen deposition and control the scarring. Then we tried to test the clinical relevance from this finding. And what we did is that we isolate fibroblasts from airway tissue collected from resected sections of the trachea in pediatric patients that had or no prior surgical history. And what we saw, it was that the number of surgeries was positively correlated with the level of activation of fibroblasts. So the more surgeries that the patient had, the higher the activation of the fibroblasts measured by the expression of CTHRC. These fibroblasts there were also producing more collagen and proliferating more, suggesting that airway reconstruction was inducing the activation of those cells, and repeated injury can detriment the function. We then ask us if surgery-induced stress was also affecting the communication of the fibroblasts with other airway cells. And we use, again, computational analysis. We use cell chat, that is a tool that allows to infer communication with one cell to another. And we saw, using this technique, that fibroblasts were basically and primarily communicating with basal cells at both time points. So we then validate these findings using functional analysis by co-culturing primary isolated human basal cells with fibroblasts that express low levels of CTHRC or fibroblasts that express high levels of CTHRC. What we saw is that when the basal cells were co-cultured with fibroblasts that express low levels of CTHRC, they were forming, as we expect, colonies that have well-defined ages in this area. However, when the basal cells were co-cultured with fibroblasts that express high CTHRC, we were not able to see the well-defined colonies, and the tissue looked more chaotic. Since light microscopy was not ideal to see those changes, we then use immunofluorescence to validate that findings. And we use specific markers for basal cells, activated basal cells, and fibroblasts. And as we saw in the previous image, when the basal cells that were co-cultured with fibroblasts that express low levels of CTHRC were forming nice colonies surrounded by fibroblasts, and activated cells just on the edges defining the colonies. However, the basal cells that were co-cultured with fibroblasts that express high CTHRC lost the colony formation, and fibroblasts were closely communicating with them. So confirming what we first saw, that the fibroblasts affect the function of the basal cells. So in summary, our studies show that ideal outcomes after airway reconstruction depend highly on the orchestrated response from fibroblasts. And if this process is deregulated, that might result in anastomotic complications like fibrosis or stenosis. I have shared with you the function of fibroblasts and how are they regulating the outcomes in airway reconstruction from a basic science standpoint. But now Dr. Debra Boyer will expand on the clinical implication of these findings. Thank you.
Excellent. Thank you, Jazmin. There you go. If you want to go to the next slide. Thank you guys for having me. I feel a little bit out of my element as I clearly don't understand the basic science as much as you all, but I do have a good clinical experience. So as mentioned, I am one of the pediatric lung transplant doctors here at Nationwide Children's Hospital in Columbus, Ohio. And so I do have a lot of experience in pediatric lung transplant. I think, the issues, and I didn't put a photo of this in here, but when we're talking about pediatric lung transplant, if you're doing a heart lung transplant, then we're talking about a tracheal anastomosis. If you are doing a bilateral sequential lung transplant, you're doing actually main stem, so right main stem, left main stem, bronchial anastomosis. And so when the majority of issues tend to be more in the bronchial anastomosis, and that's probably because that's what we do more. We tend to do more bilateral sequential lungs than we do heart lung transplants. But when you look through the literature on the incidence of airway complications, the incidence is about 2 to 30%. I guess it depends on how you define it and how they're looking at it. Why this happens and in lung transplant is that the surgeons do not re-anastomose the bronchial arteries. So the circulation to this newly anastomosed area is dependent on retrograde, very low pressure collateral flow from the pulmonary arteries. And so it's really at risk for significant complications that I'll get to. Certainly, there are other risk factors to having airway issues, including just having primary graft dysfunction, infections. A big one is Aspergillus fumigatus, as well as the use of the drug sirolimus because it inhibits wound healing. And so when that is used early, it is definitely an issue. And again, I'm using lung transplant as a clinical example. Certainly, there are other scenarios where there are airway issues. I think it's the one probably most studied, and that's why I'm focusing on it here. Next slide, please. So when we think about the airway complications that I see most commonly after lung transplant, dehiscence, so where the anastomosis kind of breaks apart a little bit, the development of granulation tissue, as well as stenosis, often related. You can get tracheo or bronchomalacia, whether you're doing a tracheal or a bronchial anastomosis, and that means really just floppiness or sort of weakness of the airway. And then certainly risks for infection as well. Next slide, please. So I wanted to show you again, this is a paper pretty recent from 2023, and it shows the cumulative incidence of airway complications after transplant. The x-axis is showing you months after the transplant, and on the y-axis would be the cumulative incidence. And I think while the number here falls in that range that we talked about before, up to 30%, so the incidence here is almost 22%. But most importantly, you can see that the majority of airway complications occur early on after transplant. Really here, it's within the first 6 months, and after that, it really does tend to reach a plateau. And that's again because the perfusion is at its lowest point. Eventually, there'll be collaterals that will develop, and so the situation does stabilize. But there really is, to Jazmin's point, a lot of stress that's going on in the airway at this point in time. Next slide, please. So I imagine that many of you are not, maybe some of you are doing bronchoscopies on a regular basis, but before I show you some abnormalities, I thought it'd be helpful to show you an image of a normal anastomosis. So this is an airway. The sutures that you see, I can point at my screen, but that probably won't help you. Thank you. So those lines that Jazmin is pointing out, those would be sutures, and you can see through this, the left main stem anastomosis, you can see through there. And you can see how it's quite patent and quite open. If we go on to the next slide, these are examples of patients of mine that this is one patient. This is both his left main stem on the left and his right main stem on the right. Again, this is a younger child. But first off, you can see those little blue lines in both images. Those are the sutures, so that's where the anastomosis occurred. And the airway should be pretty much as large as you saw on the previous slide. Instead, you can see it's really quite narrow. There's fibrosis there. It's quite stenotic. And in this particular case, it's likely that I was not able to advance a bronchoscope through his airway. Again, most of the airway issues that we see are at these anastomotic sites. If you go to the next slide, This is a different patient, but this is not at an anastomotic site. This is also a stenotic area in the lung. This is a patient's bronchus intermedius. So if you know human airway anatomy, you go into the right main stem, the upper lobe takes off. The part that continues on is called the bronchus intermedius and leads to the right middle and right lower lobes. So this is that takeoff that will lead to the right middle and right lower. And again, it should be much more open than it is here. This I would describe as pretty stenotic. This is in a patient who's pretty close to an adult size, so really should be much, much bigger. Next slide, please. I mentioned anastomotic dehiscence. So this is an image that I took from a paper referenced below. And you can see, again, the sutures there, but you can see that gap right at about 3 o'clock where there is air where it shouldn't be. And that is really a dehiscence and is really an urgent need for intervention at that point. And again, that is likely due to the poor perfusion plus minus infection in these cases. Next slide. I wanted to show you, this is a very recent paper from this year, just showing you mortality in patients who did or did not have an airway complication. So years on the x-axis and then survival probability on the y-axis. And as the blue bar are those who do not have an airway complication, the green bar are those who have any airway complication, and it could be a variety of complications. But you can see the significant difference in mortality. It does tend to level off after about a year and a half. And again, that's because the airway issues tend to happen earlier. And if the patient survives those, and it tends to level out after a while. But I have had some patients that have had recurrent airway issues, continuing on for years after their transplant. But the next summary of this slide is that airway complications are a significant contributor. In terms of treatment, I think we can do certain things, and I say we, meaning myself, along with my surgical colleagues. Certainly for things like dehiscence, it's going to be the surgeons going in there and repairing the airway, and there are lots of techniques that Tendi can probably speak more to that than I. We can put in stents, and sometimes pulmonologists or surgeons can do those. It is a challenge. There are some stents now that are removable. There also, when I first started doing this, were stents that were not removable. And that's a challenge because if you have a pediatric patient, you hope that they continue to grow, which means their airways grow and stents do not grow. So you can be then putting in a fixed stenotic area if you are unable to remove that stent. So stents can do a lot of good, but they're also not a freebie. We can do dilation of the airways where we can put in balloons into a stenotic area and inflate them. Often, though, that leads to more stress, again, back to what Jazmin was talking about, which then can lead to further fibrosis and stenosis again. So it sometimes helps, and often that is temporary. And then certainly, obviously, if it's infection, aspergillus, or other infections, we're going to treat with antibiotics, often though the damage is already done and the inciting inflammation is already happening. And even though we can clear the infection, there still can be resulting airway issues. And I think that might be my last slide. Is that correct?
That's correct.
Okay, so I mean hopefully that was helpful, just giving you some examples of finding out why this is happening and potentially ways that we can intervene would be very important for my patient population. And I'll stop there, but I'm happy to answer questions if anyone has some for me.
Thank you, Dr. Calyeca and Dr. Boyer, for the presentation. It's really a lot of work that both of you have put in, and Dr. Calyeca for a lot of exciting discussion on your part as well. I guess to start off the conversation, I found it really interesting that the perichondrial fibroblast itself lost the ability to undergo self-renewal, but then would rather differentiate into chondrocytes. Do you think that this increased likelihood for differentiation can cause the increased airway resistance that you kind of see at the very beginning, right after the surgery, especially at day 14, based on the data that you showed, Jazmin, when you found it to be most prominent?
Yes, so one of the things that we hypothesize that it's happening is that and I didn't have, I didn't put that because I didn't know if I had enough time, but basically one of the things that we saw is that surgery-induced stress was also affecting the cartilage. We have mild cartilage damage, and we believe that that was the trigger for the perichondral fibroblast to differentiate into chondrocytes. What we saw is that there is a small pool of these cells residing very close to the cartilage area, because we were able to see this structure even at homeostasis. But then the surgery was affecting the cartilage, and we believe that those are the cues that are initiating the differentiation. We don't know at which extent like this could be beneficial. When we follow the study, we follow the histological changes up to a year. And it was like showing us that at some point these perichondral fibroblasts are completely integrating into the cartilage. We don't know whether like this can be also deregulated and being bad, because we know that when that the cartilage can produce more calcium and calcified trachea, and that's also not good. But right now we don't have like a complete answer. We just know that the mild cartilage damage is triggering this response.
And
Hi,
then
Dr.
similar.
Calyeca and Dr. Boyer. Both of you, I guess, can answer this question. I'm wondering if we know if there are any types of risk stratification to kind of identify whether a patient will be predisposed to abnormal repair versus, the usual normal repair, and, how can we apply Dr. Calyeca's findings to kind of apply that to clinical applications?
I mean, I can start, Jazmin, and then if you have things to add, if that's okay. I think it's hard. I think we do have, as I mentioned, we do know patients that have some risk factors are more likely to develop anastomotic issues. I mean, in pediatrics, again, small size, because there's just not much room there. So any fibrosis that you get. So I think infants and young children are more at risk. The sicker patients are more at risk, again, probably related to just poor perfusion, of the anastomotic area, and other airways as well, which can lead to stenosis even in other areas. And then, like I said, if they have an infection, if they're having rejection, primary graft dysfunction is basically like, super early rejection. Those things happen, and that your immune system is then, sort of triggered, and can lead to more of an inflammatory response. At least that's what I see on a clinical level. I think Jazmin could probably comment on whether there's anything on a more basic level.
Yes. So one of the things that we saw, at least at the molecular and cellular level, is that the number of surgeries that the patient has was activating more the response of the fibroblasts and the cells. And some studies have shown that once these cells are activated, they are more prompt to respond. So even if the number of those cells is decreasing, if there is a second injury or repeated injury, those cells are more prompt to respond. And since those are the main producer of collagen, we at least with the spine, we hypothesize that if there are still cells that are surrounding the anastomosis and forming scar tissue, and they are not completely removed, and there is a second injury that can happen in the patient, I don't know, infection or something else, they are more prompt to respond.
So do you think there's memory in fibroblasts, and are there, I guess, indicators of this that you can detect and maybe target if that was the case? So for in cases of multiple surgeries?
Yeah, that's a very interesting question. I'm not sure of saying that they have memory, because we still don't have data that can prove that. There are a few studies that have shown similar cell types in the lung, that what basically they did is that they induced damage and then they saw the presence of those cells, and they use a model that is a model of domicing that then the animals will recover from that. So they wait until the animals recover and did a second injury. And in the animals who had a previous injury, they have like higher fibrosis areas and response, but there is still no like a specific like way of saying that it's a memory. It could be that just the cells, they have a higher proliferation capacity. So it can be that these cells are just like proliferating faster than the others. But yeah, that could be really interesting to see if it's cellular memory.
The other thing that I'll throw out there is that we have a number of patients, and it seems to be growing every year, that seem to have some immune dysregulation. Sometimes we understand it, and we've been able to really describe it on a genetic level. And sometimes we don't know, but their immune system just seems to react differently, in a almost like a hyper active state. And I don't know if that also plays into some of the things that Jazmin is describing. But again, the same patient may undergo the same things and have the same risk factors than another patient, and yet this one reacts one way and this one reacts a different way. And I don't feel like I know enough to know that. Hopefully, Jazmin and her colleagues in the lab can help us figure out how we can figure that out. But right now it's more of just, doing the best we can, and hopefully they don't develop these complications.
Yeah, I don't think that we can like fully answer that, but yeah, we saw that fibroblasts can control the immune cell response, and it could be one of the reasons why like some of the therapies are not working. The communication that we specifically saw is occurring between fibroblasts and neutrophils. But I am pretty sure that other cell types and immune cells are participating there. We still don't know how this specific subpopulation is specifically attracting neutrophils, and that's probably one of the things that we will cover soon. But definitely one of the things that we take, or one of the messages that we took from our study is that we cannot target like fibroblasts in general. They need to be like specific therapies because they have different functions, and just decreasing, for instance, the proliferation of fibroblasts can affect other functions like the immune regulation. You block the fibrosis genes, and fibroblasts can have like this immune attraction.
Yeah. And it seems like, timing is also important, right? Like when do you intervene, or when is the perfect time to intervene to kind of, revert back if it is going awry? So that's super cool.
And to build on that, I also thought it was really interesting that you saw that even though there was less ability for self-renewal, so then therefore the fibroblasts would kind of switch over and differentiate more into the chondrocytes. But then you saw that by day 60, that extra or additional fibrocartilage area had actually decreased. But according to what Dr. Boyer was presenting, she says that most of the patients come back or return to normal around 1 to 1.5 years. So do you feel like, with that said, with your other data, I believe that you actually showed that the immune response had actually gone up between day 14 to day 28, and then the fibrocartilage or the fibroblast density had actually gone down? Do you feel like there's more of a switch between fibroblasts maybe being more important at the beginning, right after surgery, and then the immune response being more important later on? Or what do you make of that?
Yes, so that's actually one of the things that it was kind of surprising, because we, for other models, we are used to see that the immune response is the primary activation, and then fibroblasts take over, and if it's dysfunctional, that can end in pathological scarring and fibrosis. The model that we use is in healthy conditions. So these animals are completely healthy, so they are like the golden standard conditions, and we believe that this is like something that needs to happen during repair. So it's not that we didn't see first immune response, we saw it, The only thing that we saw is that there is a shift in the fibroblasts. So we have first the abundance of the ones that can produce more collagen, and then start decreasing in numbers, and then the immune recruiting came, and probably just in trying to help and regulate the immune response. So there are other studies that have shown that when you block fibrosis, you can have a stronger immune response. So they are definitely tied with each other.
Niyati, you had a question?
Yeah. Hi, Jasmine. Really nice talk and very nice to see you present your work today. It's fantastic work.
Thank you.
I just had a very basic question probably. So you talked about fibroblasts being in a certain activated state, right? And then they have this entire basal cell, potentially immune cell crosstalk, a cell-to-cell communication that may mediate downstream signaling cascades and effects whatsoever. But then do you speculate that if we kind of modulate fibroblast behavior, early onset of any trigger or response that we would want to see, can it affect normal cell behavior, basically restore normal cell behavior in the future? That's my first question. And then my second question is, because you saw differential activation states of fibroblasts, did you look at it mechanistically if there was like a certain signaling pathway that was getting activated in your activated fibroblasts at all? If you could comment on that.
Yes, sure. So let me see if I understand your question correctly. So the first one is referring if we can modulate the abundance of fibroblasts as a treatment.
Yes, let me rephrase. So if you modulate fibroblast behavior, so basically not have them activated or have them locked in a transient state, would that restore normal cell behavior? Meaning, can it help us to not push them over the boundary to get into this hyper-proliferative or like that synthetic state where they are not able to behave normally? Can something like that be done early on?
Well, I don't know if that can be like directly applied into the clinic, but the functional analysis that we did in vitro was showing that we have fibroblasts that express low levels of CTHRC, that is one of the markers of fibroblast activation. The basal cells were forming normal colonies. For that specific study, we use regular fibroblasts and mouse fibroblasts, and we saw the same. They were only changing their behavior when they were co-cultured with fibroblasts that express high CTHRC. So it definitely suggests that if we are able to modulate the expression of CTHRC, we will modulate that interaction with basal cells and how basal cells are communicating with that. With the second question, yes, we did more computational analysis, and we proved that. I didn't put that data in the presentation though, but we basically saw that the communication between fibroblasts and basal cells was bidirectional. So we have like this double communication. It was not just only fibroblast to basal cell, it was also basal cell to fibroblast. And it was mainly mediated by two well-known pathways. One, it was TGF-beta, and the second one was a non-canonical Wnt. Non-canonical Wnt was exclusively fibroblast to basal cells, but basal cells to fibroblast, it was TGF-beta. So we block this pathway also, and we saw that the basal cells were, we hypothesized that the basal cells were activating also the fibroblast through this pathway. So we block it, and we saw a reduce in the activation levels of fibroblasts. But as I say, like in control conditions, you can block one pathway, but this is a TGF-beta, it's like highly expressed and regulate multiple pathways. So I don't think that it's something that you can like directly apply and expect and not expect a broad response.
Okay, yeah, thank you.
I have a sort of experimental question. In your co-culture, were you able to take the supernate and kind of like compare if there are any differences in like secreted factors, and maybe in the high expressing fibroblasts, you can identify sort of these secreted markers that also participate in the proliferative, like in the crosstalk, basically, with the basal cells?
Yeah, well, that part could be a little bit challenged because the co-culture that we did was directly putting the basal cells with the fibroblast. So if we collect the supernatant, we will collect all the general transcription factors or molecules that are secreted by both the basal cells and the fibroblasts. We won't be able to differentiate. I mean, we can have like controls in which we just have fibroblasts and we just have basal cells, but if in some way they are changing that, we won't be able to see specifically from which one are those unless we trace them in some way, maybe putting a reporter on the endoplasmatic reticulum and see which proteins are coming after that. But what we saw with the cells that they, this is not part of the study, but we know that they are changing their metabolic activity. So they are like definitely acting there. But yeah, that would be like very nice to see what specific molecule it's going.
I have a question for like just future. Like, do you plan on doing an experiment on just repeated surgical response on your animal model? Because I'd be interested to know, to also from Dr. Boyer's standpoint, on patients that require multiple surgeries, how do they respond? Do they have like an earlier immune response, more robust, or do all their anatomy of their airway, where we're going in for the surgical incisions, do they heal completely by that 1.5 years, or do you see more cartilage or, like you're saying, more fibroblast that's present there?
Yes, the answer is yes. Actually, we did. We did that experiment. First of all, I think that there are like two main things that we need to take into consideration. So the first one is that clinically the animals are performing well after a single surgery. And they can live for longer than a year. They are happily there breathing. However, we were still able to see at the cellular and molecular levels all these changes. So we need to like keep that in mind, that sometimes like probably clinical we don't have any issue, but there are changes happening in at the cellular level. With the repeated injuries, at least in our experience, didn't go well. So the animals don't tolerate repeated surgeries, and I don't know if that is because of the model and associated with that, or is something that it's also more complicated at the clinical level. So maybe Dr. Boyer can like expand more on that and saying if patients have like multiple surgeries, how complicated it is versus the one that just have like one or two.
Yeah, and it's not so much, it's rare for them to have like actual surgery and incisions and reanastomosis. What is more common is if they have stenosis, like I mentioned, we may go in and dilate the airway, and so I guess it's like surgery in the sense it's certainly a stress on the airway. And it's not uncommon, as I mentioned, in those patients to then see, stenosis reoccur after a dilation. Not always. Sometimes you're able to dilate and it stays open, but commonly, I would say patients have, get dilated and then, kind of re-stenose again after a period of time. Yes, after a year and a half, if they haven't had airway issues, the sutures can resorb, and you could almost not know in some patients that there, was an anastomotic site there, right? It reepithelializes, and you just don't really see. You would know, I guess.
I have a comment here. I just wanted to ask Dr. Boyer in the same context. So would it then differ in, elderly asthmatics, like patients who are elderly and then have asthma at the same time? Because these patients would ideally have like stiffer airways. So how would you then, imagine their airways to behave in that environment, like especially with like repeated surgical trauma and other things? Do you have any comments on that?
You're talking about a patient who may have asthma and then goes in
aged, yeah,
and then has a transplant or something.
Yep, something
yeah,
like that.
I think certainly in pediatrics and even I think in adult medicine, I don't know that there's any data that shows it's a risk factor for more airway issues or stenosis or any different behaviors. And it can go both ways, right? You could have a donor who's asthmatic or a recipient who's asthmatic, and how does that immune reaction play a role? And I don't think we know. It's certainly not something that we screen for. And again, I think the numbers are small enough that I think it's hard to say. I think there are other more likely risk factors that lead to it. But with that said, if a patient, a recipient doesn't have asthma and they receive lungs from somebody, who had asthma, then yes. But again, we don't. If a donor had significant asthma, we probably wouldn't accept those lungs for transplant, because of concern about how they behave. It's not necessarily the other way around. If a recipient has asthma, that's not so much of a problem. So that's a long-winded way of saying I'm not sure that we know anything about your specific question.
Yeah, I think that's why asthma in the elderly is kind of like a big explosion right now, because it's geriatric asthma and there are so many other, different remodeling patterns. It's just a whole different scenario right now. So Thank you. So Jazmin, did you see any sex-specific difference? I mean, it's just said out loud. Like, did you see any differences between like male versus female mice in your study, or did you not look at sex-based differences at all?
Yeah, we didn't evaluate sex as a biological feature, so we don't know exactly how it would be. Yeah, sorry.
And do you have plans to look at it? Because I think there have been established studies on that earlier, or
yeah, there are like some studies actually from our group that were conducted early on, and they were it was more sex mixed match. So the tracheal graft was from males transplanted into females, and see how there was this interaction, and in the host not to study per se the response to surgery-induced stress. This mixed match cause immune cell response, mainly driven by macrophages.
Sure.
But I'm not able to answer like specifically if they could be performing in the same sex, male graft into male host and female graft into female host, if they're going to be cellular or molecular changes.
Okay, thank you.
Jazmin, I know that with your work, I think your end goal is to try to find or develop some kind of targeted therapeutics for essentially the wound repair, tissue repair. So what specifically are you hoping to target? Is it in the early differentiation phase, or to try to improve remodeling? Or can you expand a little bit on that?
Sure. So I think that we saw these different subsets of fibroblasts, but one of the main ones that we have been like focused more is the CTHRC1 fibroblast deactivated, because they have been associated with fibrosis, not just in the airway, but in other organs and also in different diseases like cancer. And one of the things that we saw in our model that when we saw repair is that these fibroblasts were just transiently expressed. So we make us think that they need to decrease at some point in numbers. And we think that if these numbers are not decreasing and the fibroblasts remain activated, that's what is resulting in fibrosis. So we are currently conducting some studies on trying to understand what are the differences in the molecular difference in a fibroblast that is activated and expressing CTHRC versus the ones that are not. And so far, some of the data that I can share now is that they have different metabolic demands. And we try to identify which molecules specifically are regulating that. And when we block that and we inhibit the activation of this specific metabolic response, we saw that they are also more susceptible to damage. So what we are seeing is that the therapy needs to be more precise, because cells that we believe that the activation is in some way protecting the fibroblast from susceptibility and from cell death. So right now we are trying to understand that, but our focus is not using a specific drug. It's more understanding how that drug will affect the different subtypes, because that's what we're gonna see anyway in a patient. But yeah, so far we are more focused on the metabolism now.
I mean, I'm no expert in this, but I believe that the CHRC protein, right, is secreted by macrophages. So do you kind of expect some kind of concurrent macrophage response with other signaling?
Yes, this molecule it's not exclusively from fibroblast. However, it's mainly produced by fibroblast, and we haven't went into any detail of checking how that affect macrophages. But at least in our model, the cells that mainly produce CTHRC were only fibroblast, and a specific subtype, not all of them, or at least in different levels. Mm-hmm.
Thank you so much for your discussion. It's really engaging to both you and Dr. Boyer. And it was, again, a really fantastic paper with a lot of work that you put in there. So it's almost 2 o'clock. I would like to just open up again. If there's anyone who has any questions, feel free to put it into the chat or speak up at this point in time. If not, if you come up with questions later on, please feel free to email us and then we'll forward your questions along. I guess we'll go ahead and conclude here. Thank you so much for joining. Thank you so much to both you and Dr. Boyer for agreeing to be a part of this fantastic discussion. I think it was really engaging. And then if anyone else has any further questions, we'll definitely reach out to you. Thank you so much.